super resolution imaging is a part of the palette of tools. It's part of the palette of tools that has been used extensively to study the cytoskeleton, and I'd like to share with you some of the results and improvements that has been made on this subject. But first, let's introduce a bit cytoskeleton. Maybe you don't know yet what it is. So it forms the architecture of all the cells, and it's composed by 3 different filamentous networks that achieve different function in the cell, and they also display different localization. They has been differentiated first due to their difference of diameter, and we identified the microtubules, which are the widest fibers you can see in green on the image on the side. They have this, they propel from the center, from the nucleus, and go all around the cell. And the intermediate filaments that has intermediate size diameter, which are the ones that are in red here, and they are about 10 nanometer diameter. And the microfilaments of actin, which is very well known and extensively studied, and which forms a cortex all around the cell. And you can see it in blue on the image. As I said, they are involved in many cellular functions essential for cell maintaining cell shape and internal organization, and they really are very active and the motor of cell motility, cell division, the intracellular trafficking, and they also allow cells to interact with each other, within each other. So that's why this is a topic of choice, and you need to understand it very well to understand cellular processes. And one main characteristic of the cytoskeleton is its constant remodeling. It's due to the fact that it has to be involved in so many processes. And for example, here I choose to show you the behavior of the actin network of during fibroblast migration. And you can see here highlighted. So actin has been stained on these pictures, and you can see that it forms bundles that projects to allow the crawling of the cell on the surface, and then it joins back the underlying cortex of actin. So it's a highly dynamic network. We also can see here on this video the behavior of microtubule during cell division in a Hercyne embryo. And microtubules are known to organize the cell division, and they are actively participate to chromosome segregation during division, and they are really at the center of this process. And you can see here how they can change their localization, or they pull the chromosome on each side of the future daughter cells to allow the proper chromosome segregation. To study these processes and to visualize the cytoskeleton, there are many ways to do that. And for the technique that gives access to the fine structure of the cytoskeleton is electron microscopy. Here I showed you three examples of the transmission electron microscopy images of actin filaments, in the intermediate filaments and microtubules, where you can see really the difference in size of each of these networks. But the problem with electron microscopy, even if it gives access to the fine structure and of the proteins itself and the assembly of the protein, it cannot be applied to study processes in live cells, and it requires heavy sample preparation. And when you observe transmission electron microscopy images, you will see all the structures that are contrasted on your sample, not only the one you are interested in. Here, I choose to show you in vitro polymerization of proteins that has been purified first and then polymerized in vitro to be observed. That's why you have only the filaments. Otherwise, you would see a huge mesh of all the actin, intermediate filament, and microtubules together in the cell. To get rid of this unspecificity, we can use fluorescence microscopy, which gives you access to target really the protein you are interested in and stain it specifically. And one of the major advantages is if, depending on the technique you are using, you can see really the dynamics of the cellular processes and following your protein that has been stained and that will be highlighted during your observation. But it is limited by the diffraction limit of light, which narrow the resolution only to 200 nanometers, and you won't see structures that are closer or smaller than this resolution barrier. There is a way to combine, to be in between those two. Like, our goal is to achieve the highest resolution possible, but using the easy sample preparation that you can find with fluorescence microscopy, which is known to be easy to implement. And to do so, super resolution microscopy could be the perfect tool because it combines access to a high resolution with specificity and some potential for live imaging, depending on the techniques you are going to use. Again, depending on the technique, it can require specific dyes or some post-processing that can maybe be a limitation for users, but we will see this a bit later. So as I said, the goal is to reach electron microscopy level resolution. With electron microscopy, you can have a nanometric resolution, which is which can't be accessible for now with live microscopy. And despite it's a quite old technique, it is still widely used for everyday studies, independently of the field in biology. But it has many drawbacks, especially the cost and the really heavy sample preparation and imaging time, and you need to have access to electron microscopy facilities to be able to have to take your images. So it has a great advantage to give you the access to the finest detail, but not very easy to implement in a lab. And our ambition is to achieve this resolution, but with fluorescence to be specific on the sample you want to observe and in living cell. And here I showed you an example of the nuclear pore complex that has been imaged in electron microscopy and in storm on the left image. So in this case, it's not live imaging, but we can see that we can specifically see the shape and the details of the nuclear pore complex with a 20 nanometer resolution without being polluted by all the background around. I will explain a bit later some details about the storm technology. But it exists three paths to lead to a better resolution with light microscopy. First, you can make the scanning spot smaller. This is what is used in STED microscopy, where you have two laser beams that are aligned together. One, the excitation one, which gives you the same PSF as before, and another one which has this donut shape, which is depleting the signal around the center of the spots. Then, when you deplete the signal, you can give a smaller PSF and give access to a better resolution. Either you can scan with something that is not a light spot, and that's what we call interference microscopy. This is the case of SIM microscopy, where you use a pattern. You put a pattern between your light illumination and your sample, and then by moving this pattern and doing a post-imaging reconstruction, you can give access to finest details in your sample. Or you can change the sample. That's what is used in single molecule localization microscopy, and see only individual molecule, compile the many images of this single molecule imaging, and then having the super resolution image after processing. I will go a bit more into detail for each technique and show you example of how it has been used to study cytoskeleton. In the case of STED, which stands for stimulated emission depletion microscopy, we use a raster scan co-aligned with an excitation airy beam and a depletion donut, as I showed before, to leave only a sub-diffraction point spread fraction. And it gives you access to resolution around, if you push the power of the depletion beam, you can reach 20 nanometer resolution. So it has the advantage to give you a very nice resolution, not close to what we can get with electron microscopy, but you can have a direct visualization of the result of your image, because there is no post-processing step. And depending on the power of the depletion laser you are going to apply, you can reach 20 nanometer resolution, but more in everyday experiments, it's around 50 nanometer resolution, lateral resolution. And you can combine several colors, up to 2 colors, if I remember well, at the same time for the same sample, which is interesting. Despite all these advantages, it can cause photobleaching and phototoxicity, as you are going to use high power laser, and it requires the use of very stable dyes to be implemented. It has some limitation in depth, like almost all optical techniques, and due to this photobleaching and phototoxicity, it's difficult to implement for live cells, but it has been done. And for example, here in this study published in 2017, they use STED live imaging on T cells to characterize the difference in organization of the actin network during the migration of T cells. They looked at the lamellipod formation, so this projection that the cell is going to use to move on a surface or on different tissues, and they characterize the difference in structure of all the sub-network of actin depending on the position or the activation of the T cell. And for this, they performed a live observation of this rearrangement in the network at the immunological synapse with a 60 nanometer resolution. Another technique that can be used and is usually favored due to its easiness to implement is a structured illumination microscopy, and it's really well dedicated to live imaging. It's one of the best compromise between resolution and live imaging. It uses you apply a structuring pattern, across the sample field of view, and you separate, and by moving it several times, you give access to high frequency information from your images, which gives you access to the tiniest detail in the sample. It has many advantages. As I said, it's well adapted for live cell images, and it doesn't need to use specific fluorophore nor special sample preparation, so fast to implement. It allows as well fast imaging, because you are not scanning point by point your image, but having a wide field of view of your region of interest. But it requires to have very sensitive camera to catch the tiniest photons that can be displayed, emitted by your sample. It can cause as well photo bleaching and toxicity, which is always an issue when we are doing a live imaging. And the reconstruction gives a possibility to have some artifacts emerging from the processing of the images. So we have to be careful of particularly on this step, and it doesn't give access to the layers that are deep in your samples. And scene microscopy has been used in this very nice publication from 2018, where they use advanced microscopy technique in live cells to monitor the remodeling of the endoplasmic reticulum and other organelles in the cell that are closely related to microtubules and actin. Here I focus more on microtubules imaging, but I really advise you to go through this publication if you want to see very nice images of SIM live microscopy. And here, so they will highlight the interaction between the endoplasmic reticulum, which is depicted in purple, and the microtubules that are in green. And they found that during the endoplasmic reticulum extension along the microtubules, it's driven by the depolymerization of the microtubules, and they can really visualize how microtubules are driving the tubular formation of endoplasmic reticulum. In another part of their study, they focused on how. Sorry, I must come back to the fact that microtubules are involved in the motor of the intracellular trafficking. So when cell needs to move a vesicle from one side of the cell to the other, it uses the microtubules network. And here they visualize that microtubules actually form corals around the endosomes to facilitate the interaction between endoplasmic reticulum and endosome during the intracellular transport. And you can see some endosomes really moving along the microtubules and then being closely circled by endoplasmic reticulums. And by applying this microscopy technique, they have been able to catch this very transient phenomenon during intracellular trafficking with a very high resolution and accessible with other light microscopy, wide field microscopy techniques. The last big family of super resolution microscopy use instead of changing the illumination or the patterning the light, it uses properties from the sample itself which is called stochastic super resolution microscopy, and to use dyes that are either photoactivatable or photo-switchable to give access to the single position of each molecule. And by acquiring a lot of images, you can get and reconstruct a super resolution image. I show you this very, very famous video illustrating the principle of this microscopy. When you really track for the position of each molecule individually, then you compile them all to give to form back the entire structure of your sample. This technique gives you the highest possible resolution, and it allows multi-channel imaging with low illumination power, which seems initially very suitable for live imaging, but actually, as you need to multiply the number of acquisition, it can increase the acquisition times and being non-suitable for live microscopy. It also requires specific fluorophores that you need to implement before and apply to your samples. Sometimes very specific buffers that are not compatible with live imaging. And it's again limited in the depths that you can image on your sample. And in this study, for example, they use different techniques of very advanced microscopy, including single molecule imaging, and they show the improvement of each technique, for example, from wide field to SIM, where they here image raw GTPase protein that interacts with microtubules and that is that regulates the microtubule disassembly. And by looking closer and using a high advanced super resolution technique, they can actually see a differential organization of these raw GTPase that can be either surrounding the microtubules or partially interacting with microtubules. And this kind of information were not accessible with a lower resolution, as given by the SIM. In addition to these three big families of microscopy, here at BioAxial, we developed another tool that gives you access to super resolution imaging, but with gentle illumination, so that it's fully compatible with live microscopy. It's actually in taking the advantage and learning from structured illumination, confocal microscopy, and PSF engineering to take the best of these three techniques to lead to the best resolution with the lowest phototoxicity and photobleaching. And it can be used with regular fluorophores at SIM, like SIM. Instead of like SIM illuminating the whole sample with a pattern before the illumination, between the illumination and your sample, you are actually patterning your light point by point and scanning your sample. That's why it uses less light and it's less subject to phototoxicity. And it will give you access to a lateral resolution about 90 nanometers, even 70 nanometers in our latest attempts. If you want to know more about CODIM imaging, please come back to us and we will be very happy to give you more detail about the technology and all the performance it can achieve. But maybe have in mind that this can be an additional tool to add to the palette of super resolution techniques that already exist. To conclude about that, we have seen that it exists a palette of tools to explore the finest detail in biology, and we always have to make a compromise between resolution and specificity with live imaging at the same time. And super resolution microscopy allows to reach a very high resolution without having the heavy sample preparation found in electron microscopy. And it's getting always toward, it's a very active field in a constant evolution, and it's going towards direct monitoring of subcellular dynamics in real time, so more and more compatible with live imaging, as it's very often required in all fields of biology. And in the future, they are trying to combine super resolution microscopy with other optical techniques like expansion microscopy or light chip to give access to finest detail, or better, to going deeper into the sample to make the work of biologists easier and having the nicest study possible. And on the computational side, great improvements of the processing algorithms have to be expected in the future, thanks to the inputs of the artificial intelligence that we are currently living now. With this short webinar,